Okay, so this is going to be an attempt at me illustrating the process of translation. If you recall in the last pencast, the transcription process made us a piece of messenger RNA, and it was sent to the cytoplasm where it found a ribosome. If you remember from first semester, ribosome are the sites of protein synthesis. If you look at a ribosome really up close, it's kind of like it has two parts. It has two subunits, and the two subunits go together. So this is the ribosome. And this piece of messenger RNA, which is a single-stranded nucleic acid, will fit in between these two parts. So we're going to kind of put it in here, and we're just going to make up a sequence. So there's a pretty big sequence of messenger RNA. Now, one of the things about the genetic code that was discovered decades ago was that it's actually encoded in sets of 3 bases. So I'm going to underline 3 bases at a time, and those are called codons. So you will always find a codon on messenger RNA. Sometimes it's called a triplet. And in your textbook, there is a decoder chart for these codons. So it turns out that this ribosome will look at 3 bases at a time, and that will be an encoded language that stands for a certain amino acid. Now remember, an amino acid is the building block of a protein, and that's the purpose of protein synthesis, is to build these proteins. So let's assume that we've already read that the ribosome has already read this first codon, CGA. So CGA is actually an encoded language for the amino acid arginine. So if I look up CGA on this genetic code chart in your textbook, I will find that it actually stands for a, oops, misspelled that, amino acid called arginine, which we abbreviate with 3 letters, ARG. So arginine is actually attached to a really funky piece of another kind of RNA called transfer RNA. Transfer RNA. Transfer RNA hangs out in the cytoplasm. It's sometimes called tRNA. And it's shaped in a really funny shape. And all along this line, of course, are different bases, like different, let's just say C's and A's and U's and G's, okay? But down here, and I'll keep drawing some of these little bonds, the most important 3 bases are right here. This one, and this one, located at the bottom. And this is called the anticodon. And as you might guess, the anticodon is going to be opposite of a codon. It's going to be a complementary set of bases. So if we've got CGA as our first codon, then the opposite of that is going to be GCU, because remember, we're going from one type of RNA, messenger, to a second type of RNA, transfer. So there are no T's. So this anticodon of CGU will temporarily base pair with the codon of the messenger. So I'm not going to redraw this, but this is the transfer RNA that we just drew down here. And it temporarily base pairs with the codon. So anticodon, codon. Now, it's called transfer RNA for a reason. It transfers, it actually carries an amino acid. So I'm going to represent the amino acid as just a circle. And so this amino acid that's being carried by this particular transfer RNA with this particular anticodon is called arginine. So it brings arginine, ARG, to the ribosome. So now the ribosome slides down and it reads the next set of bases, UAU. And UAU is a codon for an amino acid called tyrosine. So imagine I had another transfer RNA, but down here on the anticodon, I had the opposite of UAU, which is AUA. So imagine this anticodon bonding with this codon. So up here is the codon. This anticodon, AUA, which is part of the transfer RNA, is going to transfer in an amino acid called tyrosine. So now let's let the amino acid move on down, and it reads the next codon, CCC. The anticodon for that is GGG, but we look up CCC on the decoder chart, and CCC happens to be an amino acid called proline. So the transfer RNA that has the anticodon GGG, because that's complementary to CCC, brings in proline, and it is connected to this growing chain of amino acids. The ribosome then moves down again, and it's going to read this codon, GGA, and it turns out that GGA, the opposite of that is CCU, and CCU actually translates again to proline. So another proline comes in. And then finally, the last codon in the messenger RNA strand, in this case, is UGA. And UGA, when you translate that, it doesn't actually have a complementary base sequence. It's called the stop codon. And it's the indication that this ribosome has reached the end of the messenger, thus the end of the original gene, and it's done making the protein. So there's not actually going to be a protein. There's not going to be an actual protein amino acid that we're going to connect to proline. Proline's the last one. So now this amino acid chain, which is called a polypeptide, releases from the ribosome and goes through a set of steps called folding, and it becomes a protein. So I'll illustrate this on the board. It'll be so much easier when we do it on the board together. That's it.